All right, good evening. My name is Hunter, and this is Content Review Week 4 for Principles of Biology students. Today we're going over viruses, bacteria, and archaea. So how do viruses affect host cells? By definition, viruses are obligate intracellular parasites. What does this mean? We can break this down just with the vocabulary, right? Obligate here means something has to happen. It's necessary for this organism. Intracellular, intra, inside, cellular, inside the cell, and parasites, living in or on a host organism and taking something from it without providing any benefit. Right? So an obligate intracellular parasite is one that can neither survive long term nor reproduce outside its host. The reproduction piece here is kind of important as far as how we define viruses specifically. They're not technically life forms as we would categorize other things because they don't have the machinery necessary to reproduce on their own. They require a host. So in general, how are viruses classified? Um, Viruses can often be classified based on the type of nucleic acid that they contain. So you can have single or double-stranded RNA or single or double-stranded DNA, for example. Stranded, stranded, single or double-stranded RNA, single or double-stranded DNA. Um, what is reverse transcriptase? Where was reverse transcriptase first found, and of what use is it to viruses? Of what use is it to scientists? So reverse transcriptase was discovered in retroviruses, the most famous of which being HIV. Let's see. In retroviruses. And these are any of a group of RNA viruses which insert DNA copies of their genome into the host cell in order to replicate. The enzyme that allows retroviruses to make DNA from RNA strands and then add or subtract RNA from DNA is the piece of interest here. Um, it's useful in genetic recombination to make cDNA from mRNA and avoid the problem of eukaryotic introns. Eukaryotic introns, remember, are the pieces of DNA that don't necessarily get expressed in protein. Um, Exons get expressed, introns are silent parts of the genome. Um, that was a whole lot of talking and not enough writing. So RT, reverse transcriptase, um, is the enzyme that allows retroviruses to make DNA from RNA. And then add or subtract RNA from the DNA. Useful for genetic recombination. Please don't show predictions. Thank you. All right. What use are these for scientists? Um, it's useful for genetic recombination. So some of the same reasons that it's useful for these retroviruses also make them useful for scientists in inserting new DNA into genomes and creating artificial recombinants, which is kind of cool. Why must viruses invade other cells to reproduce? Describe the general process of virus reproduction, include a discussion of lytic and lysogenic viral cycles. So I kind of already mentioned how viruses don't possess the machinery for reproduction on their own. Um, some viruses are lytic only, and some are lysogenic and lytic. Um, let's see. Mm, There we go. Take over the cell's machinery to make more viruses, and then they lyse the cell to release those viruses to allow the new virus to escape and attack other cells. Some viruses also have a lysogenic stage, and this will alternate between viruses that have a lysogenic stage will alternate between lysogenic and lytic cycles. Um, During the lysogenic stage, the virus incorporates the viral DNA into the host DNA and remains quiescent there, remains quiet, replicating with the host's DNA, and it can later come out of the host DNA and enter a lytic cycle. Some environmental conditions that stress the host cell may trigger this process. It's also what allows a lot of viruses to remain dormant in your system for a long time as they accumulate. So let's see. Which type of viruses are more likely to have a lysogenic phase? These are more likely to be double-stranded. Let's see. DNA viruses specifically, and retroviruses. Um, RNA viruses that do not produce DNA as templates for the reproduction are unable to incorporate into their host DNA. So this is the exception here, or not the exception, but this is what differentiates them. RNA viruses that do not produce DNA as templates for their reproduction are unable to incorporate into the host DNA. So usually where we go in eukaryotic cells from double-stranded DNA transcribed into messenger RNA, translated into protein. There's a difference in ordering here where RNA viruses that don't produce their own DNA templates can't then incorporate it into our DNA to then be transcribed and translated. But double-stranded DNA viruses that can produce uh, double double-stranded DNA can incorporate um, into eukaryotic DNA and have that lysogenic phase. More than 100 different viruses, many of them rhinoviruses, can cause the common cold in humans. Other viruses, influenza viruses, cause the flu. Although there are many different antibiotics for treating bacterial infections, relatively few drugs are available to treat viral infections. Explain. Um, Antibiotics used to treat bacterial infections target bacteria-specific structures or functions. These could be the 70S ribosomes, bacterial cell wall structures, or bacteria-specific enzymes. These are very, very different from the eukaryotic ribosomes, cell structures, metabolism, enzymes. And so it's easy to make antibiotics that target those specific structures and functions relative to ours. It's much more difficult to find something that a virus requires for its function that is not also required um, by the host. And that is because it uses so much of the host's uh, machinery to reproduce in its life cycle. Um, Target, let's see. It is more difficult to find. Um, the example given here is when a virus specific coat protein or enzyme is found, such as reverse transcriptase. Um, you also have the issue of high rates of mutation that make it difficult to develop a drug or vaccine, um, or other measures against it. Um, COVID is the example that I used last year. We used a coat protein to develop an mRNA vaccine that taught our cells to create those coat proteins, which are harmless to us, to recognize it so that we could build defenses against it. But then, as COVID kept circulating in the population, it mutated to subvert those defenses. Those coat proteins changed. And we don't have, well, we can't, we don't have time to get into the specifics of the immunology of that. But that is why, right? These viruses mutate very, very quickly. Um, so high rates of mutation make it difficult to develop drug or vaccines against it. So even when we find something, it tends to change very, very quickly. Develop a drug combination to effectively combat multiple infections. Patients with AIDS often acquire opportunistic infections. Opportunistic infections are ones that take advantage of the suppressed immune system caused by HIV/AIDS. Um, Imagine a patient with AIDS and two opportunistic infections, a respiratory disease caused by mycobacterium and another disease caused by the fungus Candida. As the patient's physician, you need to prescribe drugs to counteract the infections. Given the normal anatomical and functional characteristics of each disease organism, what characteristics should a drug combination have to treat each of these three infections while doing the least harm to the patient? There are lots of ways to answer this question, but we need to basically rehash a little bit of what we've been talking about already and address them separately. So first we have, let's address the AIDS. This patient will require continual treatment using several different kinds of drugs because the virus will eventually adapt or evolve to tolerate most treatments. But these might be antiretrovirals, among their other therapies. The mycobacterium we can treat with antibiotics specific to this bacteria. The best option for treating this particular one would not be penicillin, for example, because while penicillin compromises the peptidoglycan wall of many bacteria, mycobacterium is not affected by penicillin. So we're going to need to use something like erythromycin. Erythromycin. And this interferes with bacterial protein synthesis specifically by affecting the 70S ribosomes of bacteria, which are different from the 80S ribosomes in eukaryotes. Finally, for Candida, Candida is a very common fungal infection. And like humans, yeasts are eukaryotes, which makes them particularly difficult to treat. So, given the many similarities in eukaryotic cells' structure and function, the fungal infection is more difficult to treat compared to the bacterial infection. However, it may be possible to target the fungal infection, target the fungal cell walls because humans don't have cell walls. Um, it may also be possible to destroy the fungus exoenzymes that are needed to break down nutrients, so exo outside um, enzymes. So target cell wall or exoenzymes. Enzymes. Um, Athlete's foot is caused by a very common bacteria, or excuse me, a common fungus called Tinea corpus. and is treatable by a lot of over-the-counter um, antifungal treatments like clotrimazole or terbinafine. Um, and you can get some prescription antifungals that'll be Peros as a in pill form that will do a more system-wide treatment. But they are pretty tough to treat. What are the main characteristics of bacteria and archaea? Based on DNA sequence analysis, three major domains of life have been proposed. What are the three major domains of life? What sets of characteristics place organisms into one domain versus another? We've kind of been talking around this. We've got bacteria, archaea, and eukarya. And while bacteria and archaea share some characteristics, I have it in my head that they're actually archaea are more closely related to eukarya than to bacteria. Alrighty, so bacteria have prokaryote cell structure. Don't need that. Uh, most have circular chromosomes. We have cell walls containing peptidoglycan with muramic acid. Muramic acid occurs naturally as N-acetylmuramic acid in peptidoglycan. Um, all bacterial species use the same type of RNA polymerase, one type of RNA polymerase. And bacteria use formal methionine as their initiator amino acid. In order to start protein synthesis. So a formal group has been added to the amino group and is specifically used for initiation of protein synthesis from bacterial or organelle genes. So for archaea also have a prokaryote cell structure, but archaeal cell walls do not contain cell walls. Peptidoglycan or muramic acid. They also have circular chromosomes. And they have several types of RNA polymerases specifically, sorry. And these can be found among the different archaeal species. And archaea use methionine as initiator amino acid. Finally, eukarya. Eukaryotes have the eukaryotic cell structure with individual compartments and a nucleus and so on and so forth. So if you see something that looks like a mitochondria, it's probably eukaryotic in origin. Um, wow, we're floating right off the page here. Eukaryotes have linear chromosomes. Eukaryotic cell walls, when present, such as around plant cells, generally contain cellulose or chitin. And methionine, just as with archaea, is the initiator amino acid in translation. Yeah. Bacteria are divided into 6 major groups. What characteristics are used to place organisms into each of these groups? Each of these have kind of unique clinical presentations. They also have a lot of utility as far as what we can do with them. Some of these are good for making yogurt and cheese and um, as soil decomposers. Some fix nitrogen. So it's easy to think of, like germs bad, but they provide a lot of utility as well. And they reproduce really, really quickly and have many, many generations very in a short time span, which make them really great um, test organisms. So firmicutes, I think these are all Latin in origin, but firmi, meaning hard, literally, and cute, think cutaneous, like skin. This literally means it has a solid or a strong wall, which gram-positive is another way of saying the same thing. Um, DNA contains low percentage of guanine and cytosine. They are rod or sphere-shaped. Some fix nitrogen. Some photosynthesize. Um, some metabolize via fermentation. Uh, some via cell respiration, a lot like us. Members of this group cause anthrax, botulism, tetanus. If you've ever eaten expired, if you've ever eaten expired canned food or canned food where the seal has been cracked for a while, that's a good way to catch botulism. It's a really, really nasty infection. Some are used to ferment milk products. Spiroclase, so named for their distinctive spiral or corkscrew shape. Metabolized via fermentation. Some fix nitrogen. Um, syphilis and Lyme disease are both caused by these. Many are anaerobic. Given the choice between an open ulcerous wound or a cyst, one that is deep in the tissue, I would rather have a ulcer because in the cyst, the bacteria can displace enough of your own tissue and create an anoxic environment, really, really friendly to anaerobes, like these spirocales, which can be particularly nasty because they produce a lot of uh, byproducts that are super toxic to eukaryotes like us. Actinobacteria. These have DNA that contain high percentage of guanine and cytosine. These are also gram-positive, having that thick outer wall. Rod and filamentous shape. These are heterotrophic, so they eat others, and parasitic species. These are also the source of many antibiotics. So the heterotrophic and parasitic nature of these makes them good candidates for killing other bacterial infections, because bacteria would presumably be one of their preferred food items. Tuberculosis. Tuberculosis. And leprosy. And I actually grabbed this quote here. Um, Tuberculosis is generally curable. It is the world's deadliest infectious disease. In 2022, tuberculosis killed 1.3 million people, according to the World Health Organization, more than COVID-19 or malaria or HIV. And it's usually about accessibility to treatment. Uh, they're also abundant soil decomposers. Chlamydiales. These are spherical shaped and very small. All known species are parasitic and live inside host cells, causes the STD chlamydia, hence the name. And it's the most common cause, which is the most common cause of blindness in humans. Cyanobacteria have both solitary and colonial forms. Solitary, living individually by itself, it's just one little cyanobacteria, and colonial, all of them living mutualistically together, often appearing to be a single organ, uh, often appearing to be a single organism. So, yeah, dominant mass of aquatic environments. I presume they mean dominant biomass of aquatic environments. All are photosynthetic. Some fix nitrogen. Some live in association with fungi called lichens. So lichens are this mutualistic organism of a fungus living in association with the cyanobacteria, which is photosynthesizing and providing energy to uh, the fungus. Cyanobacteria are also the it is theorized that cyanobacteria are responsible for the oxygenation of early Earth's atmosphere. Um, when I was at Colorado College, I took a historical geology class that changed my life. It was super cool. We drove all over the Western United States looking at fossil evidence of um, early Earth history. And we got to see the stromatolites, which are the fossils left behind from cyanobacteria colonies, basically, the oldest fossils of living organisms on Earth. And this explosion of photosynthetic bacteria um, may be what was responsible for photosynthesizing and creating sufficient oxygen to support, as we were talking about last week, the aerobic uh, the aerobic organisms that followed. Cyanobacteria predate plants, and we owe them a lot. All right, proteobacteria has 5 major subgroups. These can be rod, sphere, or spiral shaped. They have wide metabolic diversity. Cause Legionnaire's disease, um, cholera, dysentery, all of the things you would die of on the Oregon Trail, ulcers, diarrhea, etc. They're also used to produce vinegar. And the genus Rhizobium live in or on plant roots and fix nitrogen. So super important. What kinds of arguments or evidence would you use to support the idea that bacteria are more highly evolved? And this comes with a Massive disclaimer. Um, and it depends on how we're defining highly evolved, how we're qualifying that. If we consider organisms that are closely fit to their environment by metabolism, for example, they are extremely successful in that environment. And by successful, we mean successful at passing on their genetic material to the next generation and persisting. Um, if they're super fit to their environment, and we consider that to be more highly evolved, then many of the species of bacteria are more highly evolved than humans. How would you support the idea that humans are more highly evolved? Um, you could say that the changes in morphology that humans enjoy that allow us to succeed in a variety of environments within a sort of narrow range of temperatures and pH and all that may actually make us more highly evolved. Our flexibility in that way. So, um, right, so a lot of this is subjective, and speciation is another great example where one definition, such as the ability to uh, breed with other members of its group, defines a species, um, can break down at a certain point. Like what level of genetic closeness uh, defines speciation. Some of this is very subjective, and understanding those different perspectives will allow you to sort of make informed and accurate statements about them. So given what you know about the origin of life on Earth, you want to look for life on other planets. This is a cool thought exercise. What characteristics of the planet's environment would you look for and explain your reasoning? So in my lifetime, we went from believing pretty firmly that there was absolutely no water anywhere else in our solar system besides on Earth. And now we have confirmed that there is water on Mars and there's water on Europa and there might even be water a couple other places. But water was is one of those things. Um, we know that life can survive without oxygen, but as far as we know, uh, water's pretty important. What kind of life would you look for and explain your reasoning? Um, to the best of our knowledge, free oxygen is not present on other planets. Not even most on other planets. It would make sense to look for anaerobic, uh, perhaps archaeal species. What tests or probes would you use to find the kind of life you proposed in part A and explain your reasoning? Again, there's no right answer for the previous questions, but your answer for this one should make sense given whatever example you put here. So if we make the assumption that life on other planets followed a similar evolutionary path to that of life on Earth, um, we would want to look for similar markers, um, such as life forms that have DNA or RNA as their genetic material. Um, um, yeah, we could also look for other compounds that we know are common to life, such as carbon polymers. Um, we could analyze soil for the, you know, carbohydrates, proteins, lipids, those biomolecules. I read an article not too long ago about atmospheric markers of industrialization. CFCs is a big one. Um, NO2 would be another, where if present, these would be really, really strong indicators that there is an industrial civilization on, that planet. And because those compounds don't exist um, outside of as byproducts of industrial processes. Um, NO2 and some, phosphorus compounds, which are common uh, in our post-industrial atmosphere, I think can also be created by volcanic eruptions. So you kind of have to be careful. But CFCs are one that, like, if you find evidence of CFCs, um, there is industry happening. So, all right, that is all I have for you this week. Enjoy your weekend. I will. Don't forget to turn in your worksheets. I will see you next week.